was mostly performed by the people from our lab. Here are the full set of people in our lab. Those who contributed mostly to the work I'm going to present are highlighted in red, and the people highlighted in blue have either presented already their work or will present later on during this conference. I will be mentioning specific people throughout the presentation. Now, it is a fact of life and nature that atoms form molecules. That's a given. It is also a fact of nature that molecules often tend to aggregate. Organic molecules of various shapes and sizes can cluster up, forming clusters, chains, oligomers, globules, micelles, nanotubes, you name it. The aggregates can have also different shapes and sizes. Now, what is clear from this fact alone, that aggregated states are somehow beneficial. They're usually held by some sort of non-covalent interactions, because if it will be a clearly covalent interaction, we would not call it and associate with, rather call it a molecule. Now, it's also clear that these aggregates live long enough to be detected. Otherwise, we will not talk about them. One can even say that the human body as the whole is the sort of non-covalently bound aggregate, however heterogeneous, of molecules. And if in a thought experiment, we will switch off all the hydrogen bonds in the human body, mostly formed by water molecules, which would kind of require a lot of energy though, the human body will turn into, well, not even a puddle of water like in this famous scene from the Amelie movie, but rather to a gas. It will be equivalent to an explosion. About 10 kilograms of TNT, according to my estimations. Anyway, so the type of non-covalent interactions I'm going to talk about is hydrogen bonds. Now, if we take a molecule and give it a proton donating spot and proton accepting spot on opposite sides of the molecule, then such molecules will be able to form chains. Now, if we take a different molecule and give it a proton donating spot and proton accepting spot on the same side of the molecule, these molecules will be able to form cyclic dimers. And also they will be able to form infinite chains. Now, yet again, if we take yet another molecule and give it a proton donating and proton accepting spots kind of at an angle, then these molecules would be able to form more complicated self-aggregates, oligomers, clusters, and they will be able probably to form infinite chains. Now, the most simple case of this kind of molecule is given by carboxylic acids, well known, I guess, to absolutely everyone in this audience. Here is an example of the carboxylic acid molecule. It has a proton donating OH and a proton accepting carbonyl bond. Now, here are the highlighted proton donating and proton accepting spots in this molecule. They are kind of directed in the same, they're located on the same side of the molecule. So these carboxylic acid molecules, they are able to do one of two things. They can either form infinite chains, and two carboxylic acids do that, formic acid and acetic acid. And also they can form cyclic dimers. Basically, all other carboxylic acids in crystal state form cyclic dimers. The formic and acetic acid, which form chain in crystal state, they also can form cyclic dimers, and they do it in a gas phase or in solution. This is kind of the scientific consensus, and this is no surprises here. Somewhat more complicated case is given by the phosphenic acids that contain POH and PO bonds, phosphoric acids that basically contain the same fragment but two more oxygens at the phosphorus atom, and arsenic acids where there is oxygen, arsenic, oxygen H group. The proton donating and proton accepting abilities of these molecules do not look at the same direction. They are no longer located perfectly on the same side of the molecule. Now, the locations of these proton donating and accepting spots can be visualized if one uses the electron localization function, kind of a computational methods to visualize lone pairs. And one can see, let's take, for example, the phosphoric acid dimethyl phosphate shown in the middle, that first of all, the OPOH group is no longer planar, kind of the proton looks slightly to the side of this plane OPO. And the proton accepting spots in this molecule are no longer located in the same plane. They're kind of forming a circle, a donut of possible directions of proton accepting ability around this oxygen atom. Something similar happens for the other molecules on this slide as well. So what do these molecules do in crystal state? Now, we can visualize the proton donating and accepting abilities as a sort of a cones of preferential directions of forming hydrogen bonds. A cone around oxygen, in case of phosphinic acids, a cone around oxygen in arsenic acids, and slightly differently looking cones of possible directions of proton donating ability. Now, let's start with phosphinic acids. How do they crystallize? It all depends on the size of substituents. If the substituents on the phosphinic acid are small enough, like 2 methyls in this case, then it forms infinite chains of various configurations, but topologically that's an infinite chain. Substituents such as methyl and phenyl, 2 phenyls, they're small enough, and in all cases the molecules crystallize as chains. If the substituents get large enough, even by certain amount, like in butyl groups or a bit more complicated substituents in this middle case, then the molecules start to form cyclic dimers. They're a bit skewed, they're not perfectly planar, but they are cyclic dimers. Sometimes this increase in the size of substituents, which tips over the molecules from chains to dimers, is not too obvious to a naked eye. For example, compare these 2 molecules shown in the top of the slide. They kind of similar. There's a 5-membered ring at the phosphorus atom and a couple of 6-membered rings on the sides. I would even say, just by looking at it, that the left molecule has larger substituents, but well, apparently that's not the case, and the crystal packing reasons lead to the following fact: the left molecule form chains, the right molecule already form cyclic dimers. So I would visualize it like this: if the substituents are large on the molecule that contains the proton donating and proton accepting spots more or less on the same side of the molecule, then it does not prohibit the molecule to form cyclic dimers, but it kind of prohibits it from forming infinite chains. So dimers are fine. Now, when you try to form a chain, the 3rd molecule just doesn't fit there. The substituents are on the way, so the chains are not being formed. Phosphoric acids are rarely known. The crystal structures of phosphoric acids are few and far between. Now, a couple of days ago, Artyom Yakubenko has successfully crystallized hydrate of diphenyl phosphate, the molecule that looks like a bee. This is the structure that he has obtained. Well, I'm sorry, that's a wrong picture. I probably have made a mistake. This is the structure that he has obtained. So the crystal look like layers of hydrogen bonded hexagons in which acid molecules, and let me highlight one of the acid molecules, are interlaced with protonated water dimers. Now, in this acid molecule highlighted in white, I have removed the phenyl substituents just for the visual clarity, but if I would add them, it would look indeed like a bee in a honeycomb. So in this case, there are many more bees than in the actual photo, but the structure is somewhat similar nonetheless. Arsenic acids are even trickier case because here some of the molecules show polymorphism. They can crystallize in the same molecule can crystallize either as cyclic dimer or infinite chains. This is an example of dimethyl arsenic acid or cacodylic acid. I have to mention that it is not crocodilic acid, it is cacodylic acid, and our participants from Greece, I'm not sure if they are online, would confirm that nothing that smells good would be called cacodylic. It comes from the bad smell of many arsenic-containing compounds, cacodyls in general. Okay, so this molecule, depending on the conditions, on the solvent from which the crystals are grown, can form either dimers or infinite chains. So there's more flexibility in the proton donating and accepting directions of this molecule. Okay, so this is the whole story was about the crystal state. But what happens in liquids? And this is what we are mostly interested in. If we put the molecules in solution, surrounding them by relatively inert organic solvent, how do they cluster up? What kind of complexes they do form? Here, we have to rely mostly on indirect methods, as crystallography will no longer be applicable. So spectroscopy, and of course, NMR spectroscopy is the one I will be talking about, is quite useful here. Now, one of the ways to establish at least the stoichiometry of the aggregate can be described like this. This is not the method we have used in the work I'm going to present, but I'm showing it for the kind of completeness. It can be shown on the example of carboxylic acids, which we all know already they tend to form cyclic dimers, but still, here's the idea. One can estimate the aggregation number of this molecule by dividing the volume of the complex by the volume of the monomer. And these volumes in turn can be estimated through the diffusion coefficients. So the cube of the ratio of diffusion coefficients is related to the aggregation number within all the approximations done in this reasoning. So the acetic acid in solution in chloroform, let's say, can be either monomeric or dimeric. And there is an equilibrium between these two states. So what the kind of any spectroscopic observable in the proton spectrum sees is the average, or weighted average, between the monomer and dimer. So the normalized diffusion coefficient changes when the concentration of the acid increases. So we go from very low concentration to higher concentration, even higher concentration, and keep adding acetic acid into the solution. And the aggregation number, which at low concentration is very close to 1, keep increasing, sort of asymptotically approaching the value 2. And for liquid acetic acid, it is even slightly higher than 2, because the structure of the liquid acid is kind of similar to the structure of the crystal of acetic acid, where it is chains. So we can see kind of a different chains of different length being formed slowly in the liquid acetic acid. Okay, but that's not the methods of choice for the examples I'm going to present. Now, if we have a molecule, let's say molecule A, that can aggregate into sort of self-associate, this reaction can be described by the rate constant of the forward and backward reaction. And if I would like to see in the NMR spectrum or in any other NMR observable, the intrinsic properties of an aggregate, I would like to achieve two things. First of all, to slow down the exchange between the monomers and the aggregates. So I would like to decrease both the forward and the backward reaction constants. And also, I would like to shift the equilibrium as much as I can towards complexation. So my whole data will come from self-associates. The equilibrium constant is just the ratio of the rate constants. So I would like it to be large to shift the equilibrium towards complexation. Both of these goals can be achieved by just lowering the temperature. But by lowering the temperature, I can easily freeze the whole thing. And I would like to have the high resolution spectra. So in order to overcome this obstacle, we have used the liquefied gases as solvents. At room temperature, these fluorinated methanes, or fluorinated and chlorinated methanes, they are gases. But at low temperature, they become liquid, and this liquid can serve as the convenient solvent for the low temperature measurements. Now, the gas pressure can be at room temperature can be quite high. So in order to store the samples, we have to go through the kind of a jump through hoops to prepare, to synthesize these deuterated gases, to handle them in a high vacuum system. And then eventually we have to learn how to prepare samples using the sample tubes made, precision sample tubes made of the thick glass, so they will not explode under the pressure of gases inside. These thick wall sample tubes can withstand pressure up to 7 bar. And then we have to cool down the sample area to the desired temperature. In our case, it's about 100 Kelvin. And for this, we use the standard approaches available on almost any NMR spectrometer. Okay, so second trick that we have used is the partial HD substitution. Consider this schematically drawn cyclic dimer of a particular acid. Now, there are two protons which are perfectly equivalent and give rise to one signal in the NMR spectrum. Now, if I change one of the protons for deuteron, I would change the geometry of my dimer, and the remaining proton will resonate at a different chemical shift. So this effect is called the isotope effect, the HD isotope effect. So appearance of the second signal can be the signature of a cyclic dimer. And here is an example of acetic acid. I consider this case perfectly kind of clear, that it forms cyclic dimers. Acetic acid gives rise to a single signal at low temperature, 110 Kelvin. After partial deuteration, a new signal appears at slightly higher field. Okay, so what happens if we take not acetic acid, which is clear, but phosphinic acid or phosphoric acid, which is less clear, or in this case, arsenic acid? The same happens. There is one signal of non-deuterated species, and the second signal grows and appears to the right of it, and the intensity of the second signal is exactly equal to the deuteration ratio of the sample. There is 70% of protons substituted by deuterons. There is a 17% intensity of this new peak with respect to the original one. So arsenic acids seem to form cyclic dimers almost like acetic acid. At least that's the dominating species in the solution. If we take phosphoric acid, then already without deuteration, we see two signals. There are two different complexes that these acids can form in solution. One of the complexes that give rise to the right-hand signal splits into two after partial deuteration, exactly like the cyclic dimer should. So we assign it to a dimer. The second signal, at slightly lower field, splits into three. And these larger number of isotope effects tells us that there are more spots for deuteration in the complex. We can actually count the number of spots for deuteration just by counting the number of new lines appearing in the spectrum. In this case, we're sure that we're looking at a trimer, because the trimer contains 3 protons and it can have more isotopologues. All 3 protons, 2 protons and 1 deuteron, 2 deuteron and 1 proton. If there would be 3 deuterons, it just would not contribute to the proton spectrum. So counting the number of new lines, we can count the number of hydrogen bonds in self-associate, and that's how we can distinguish dimers and trimers. That's a kind of a key point here in this presentation. Other phosphinic and phosphoric acids also form dimers and trimers with a high tendency to form trimers. So that's how we can visualize it. The carboxylic acid, the simple case, look like a Lego piece that is tuned to form cyclic dimers. It has 1 pin for the proton donating site and 1 dip for the proton accepting site, so it's quite easily to form cyclic dimers with this Lego piece. Now, the phosphinic acids, they look like a Lego piece, which Lego does not produce such a weird shapes, but here the proton donating ability and the proton accepting ability, they are located at a weird angle. So formation of a dimer is not as preferential, but formation of a trimer can be done. So these 2 molecules approaching each other leave a gap between them, the gap that can be bridged by the 3rd molecule. Just everything seems to be right in phosphinic and phosphoric acids. The angles of the proton donating and accepting abilities, the size of the whole thing just fits for trimerization. And it's different for arsenic acids that, I remind you, form mostly dimers. Technically, we have a very similar situation. It's a weird Lego piece where proton donating and proton accepting abilities are at a certain angle. But these angles are not as extreme as in case of phosphinic acids, and it's still okay to form cyclic dimers. It can still be kind of, these 2 pieces can be jammed one into another for a stable cyclic dimer, which is harder to do for the phosphinic and phosphoric acids shown in the middle. Now, if we mix 2 different acids, like 2 different phosphoric acids, there are many more signals in the proton spectrum. Some of them are known already to us because we have studied the individual acids. So some of them come from self-associates of the acids. All the other signals come from mixed complexes. And we can deuterate the sample, and the number of signals increase even further, but actually the ability to understand what's happening increases. Because knowing how the isotope effects are being formed, we can now analyze this spectrum, finding out, first of all, everything that we already know for the self-associates. These lines are clear, the ones which are marked by the vertical gray lines. But the remaining lines can be grouped into several sets. One subspectrum comes from the mixed dimer, where 2 different molecules form cyclic dimer. 7 lines, sounds like Lord of the Rings, and 7 lines were given to the trimer. So 7 lines are given to the trimer where 2 acids with the phenyl substituents and 1 molecule with the methyl substituents form a cyclic trimer. 7 more lines come from a different cyclic trimer where the 2 molecules have methyl substituents and 1 molecule has phenyl substituents. So it seems that if we mix 2 different molecules of phosphinic or phosphoric acids, they can mix and match in all combinations. They have similar shapes and sizes. It's like Lego pieces of different color, You can still match them. The color is not that important. If the substituents are not large enough and there are no steric problems, then you still can join the Lego pieces in cyclic trimers. But there are some signals that are hard to understand, what was hard to understand. In the mixture of phosphinic and phosphoric acids, sometimes there are signals that cannot be attributed to any of the complexes we have considered before. And these signals are split into many, many lines after partial deuteration. But the logic and just kind of the configurational reasoning allows us to assign these lines. We know for sure that if we name one molecule A and one molecule B, the new signal cannot be self-associates of A or self-associates of B, because we know these signals already. We have also found all kinds of mixed dimers and trimers. It cannot be those. It cannot be oligomers of higher order of individual molecules because they are not being formed. We have established it for individual molecules already. The next simplest complex that this new signal can come from are actually mixed tetramers. Trimerization already is a rare kind of self-association in dilute solutions. Tetramerization is even more rare, but I think we can prove that this is one single little signal mark here with the red dot comes from the mixed tetramer, because there are 4 kinds of tetramers with 2 kinds of molecules. Let's say A and B. 3 molecules A, 1 molecule B, 2 molecules A and 2 molecules B in different configuration, like alternating or 2 and then 2 of the other, and 3 molecules of B and 1 molecule of A. Just from this picture alone, we can predict how many signals of chemically non-equivalent protons and phosphorus these complexes should give in the NMR spectrum. We can also predict how many isotopologues of chemically non-equivalent protons in isotopologues of these molecules should be there. And they're all different. So the one we are looking at perfectly matches just one of the tetramers. 1 proton signal, 2 phosphorus signals of equal intensity, 7 isotopologues. So we assign it to this tetramer of phosphinic and phosphoric acids. There are some other cases where we found also the tetramers of not alternating molecules, but just forming this kind of 2x2 cycle. And the last case I'm going to present are phosphonic acids. These acids have 2 OH groups and 1 phosphenoxide group. Now, here is a 3-dimensional structure of terbutyl phosphonic acid. We can add the cones of proton donating and accepting ability. So we see the number of possibilities here is increased, and one could expect well, I didn't know what to expect from this molecule. How would it self-associate? In crystal state, phosphonic acid form several different structures. They can join each other in sort of an infinite chain held together by pairs of hydrogen bonds. They can form into the 2-dimensional layer where each molecule is held to several partners forming a sheet of hydrogen bonds. They can form sort of a spiral columns held together by triple hydrogen bonds. So they can form different things. But what do they form in solution? Here is a proton and phosphorus spectra measured by Ivan Giba. And there are several facts that I have listed here in red, which can be derived from these spectra alone. Whatever the complex is, all protons have to be hydrogen bonded, all phosphorus have to be equivalent. There necessarily should be 2 types of hydrogen bonds with equal number of protons involved in each type, and each phosphorus atom should be bound, should have the spin coupling to both types of protons. So it's kind of a limitations. It's a set of limitations to our fantasy of what could be formed. And there is one particular structure that fits all of them. one and only one. We were unable, and I think nobody will be able to find a different structure that fits all these criteria. And this structure is three-dimensional tetramer. That's how I think it is formed. There are 4 molecules of phosphonic acid. 2 of these molecules come close to each other and form a dimer. Dimer with 2 dangling OH protons. 2 of them are in hydrogen bond, but 2 are dangling at free at the moment. 2 other molecules come together and form yet another cyclic dimer with 2 dangling protons. Now, these 2 dimers can approach each other and form a sort of a cage, a cluster of 4 molecules in a sort of tetrahedral configuration. If I add here substituents, there will be just more atoms and harder to see. The structure of this tetrahedral tetramer can be visualized as the paper cutout. So if one prints out this slide, takes the scissors, cuts out this shape, one can fold a tetrahedron, which will show the structure of this tetramer. In the vertices of this tetrahedron, there are phosphorus atoms, and each face, each side, is formed by a couple of hydrogen bonds, chemically non-equivalent hydrogen bonds. It is sort of a forming of a tetrahedron out of 4 identical pieces. It's possible to do. There are congruent shapes that can form tetrahedron. These 2 shapes can come close to each other, form one pair, another pair, and the whole thing can join into a perfect tetrahedron. Now, with a little bit of self-promotion with this story we got on the cover of the Symmetry journal, which we're very happy about. So I think this is story has been published already. Okay. So here is my 3 main conclusions that I want to drive home with this presentation. First of all, in self-associates of organic acids, hydrogen bonds interact with each other, and the reason for this interaction is the conjugation of bonds in between proton donating and proton accepting sites. Whatever happens with the proton donor changes the electronic structure of the molecule, however small, and this change is propagated through the conjugation bonds and changes the proton accepting site, affecting the neighboring hydrogen bonds. So all hydrogen bonds are kind of interacting, talking to each other, feeling each other. This can be used in HD substitution. HD substitution is a way to slightly, softly change one of the bonds, and all the others start to react on this small change without destroying the complex. Just a little bit change one of the hydrogen bonds. All the others respond, and this new, slightly changed bonds give rise to different signals. And by counting the number of signals, you can count the number of responses. So to count the number of possible spots for deuteration. You kind of do the inventory of your bonds by poking each one of them by deuteration. Symmetry considerations can further limit the number of possibilities. Sometimes, you have to take into account that the whole thing, the left and right side of the complex are identical. That's why, symmetry considerations, I guess everyone understands. So with this couple of tricks up our sleeve, we were able to prove the structures of cyclic dimers, trimers, tetramers, and 3-dimensional tetramers of small organic acids: carboxylic, phosphinic, phosphonic, arsenic. And I guess with this, I would like to thank you for your attention. That's all I wanted to say. I leave you with this slide. There are some conferences in St. Petersburg University that are going to be held in September. If anyone is interested, please check it out. So thank you very much for your attention. I will be happy to answer any questions that you might have.
Thank you for Professor Tolstoy for the beautiful lecture. Firstly, humor comments from me. Several years ago, I have heard a lecture of Professor Newcomb, and he showed how we can create different letters of alphabet. For instance, K, X, W, and it seems you went further and show how we can play in nano-Lego.
Yeah, well, or in the terms of Professor Newcomb, mostly we're forming the letter O of different sizes. Yeah.
Okay, there's some questions, comments. Professor Belevich, please.
Thank you, Peter, for nice fundamental work on hydrogen bonding. Now it's not often can listen to such things because it's very difficult to win the project studying hydrogen bonding. My question about phosphonic acids. These substances are well known protonic conductors. But in your case, it can be, as I can imagine, you close aggregates without exchange between neighboring aggregates because of cryosolvent and very low temperature. And no effect of proton conductivity are seen. Is it so?
That's true. The tetramers that are being formed in our experiments are quite rigid and robust structures that do not exchange so easily with the other molecules. However, in the materials which use the phosphonic group for proton conductivity, the relative orientation of this phosphonic group is restricted by usually a polymeric chain on which they are located. So they are not able to come close and form this self-sufficient tetramer. They are rather spread out on a line, and then it allows the proton conductivity along the chain of such materials. This is like a kind of proton conducting wire in some cases, but in the other materials, people argue that the actual mechanism of proton conductivity are the protonated water molecules that kind of drive through the channel created by the phosphonic acid. So there are still arguments about that. But I guess our case, we just allow too much freedom of the molecules. They can now find each other and be happy with it.
Thank you.
Okay, other questions, comment, please. Okay, there's small question from me. It is, I understand that you need the low temperature to kill the fast exchanges, but maybe it is possible to use this experience or methods for study molecular system in solvent and more close to room temperature.
Well, I guess by increasing the strength of the non-covalent interaction that hold together aggregates, one can make easier restrictions on the temperature. That's true. However, I think that if we are talking about small molecule in solution and the energies of bonds that hold them together are sort of, I don't know, 10, 20 kilocalorie per mole, and the room temperature being what it is, it would still lead to enough events of breaking and reformation of the complexes if there will be small molecules in solution. So I guess, like in the question of Professor Balevich's, one would need then to restrict the motion of the molecule somehow in order to increase the lifetime of a species that one wants to observe. Attach them to a polymer or somehow restrict the motion. Does that answer your question? No, not.
Thanks for your answer. Okay, maybe more questions. Anyway, we have the time limit. Many thanks for Professor Tolstoy lecture again. Now we can continue. We continue our session. Next speaker, Valery Karpov from Saint-Petersburg State University.